Thank you, everyone. Thank you for the thank you to the organizer for the very kind introduction. Appreciate everyone taking time out of your busy morning, or if you're like me, your busy evening, to go ahead and to attend today's webinar. So again, I'll just very briefly reintroduce myself. My name is Ray Miller. I'm a Senior Global Product Manager with GenScript. I'm in charge of commercialization and product development as it relates to our peptide, our CRISPR, and our NGS service lines. As you can see from my title slide, we're going to be talking about neoantigen peptide production, making the difficult easy. To kind of give everyone a little bit of a context to this talk and kind of how we got started on this. So GenScript is, of course, a major worldwide CRO. We're, of course, very well known for our gene synthesis, our plasmid DNA. But we've been in the peptide business for, at this point, almost 10 years. And over the last few years, we've taken on a series of projects involved in the synthesis of peptides that are used in neoantigen therapeutic research and development. It's a really, really exciting area of immunotherapy. I think there's a lot of potential here. And so what I'm going to do in today's talk is I'm going to talk to you a little bit about kind of the science behind neoantigens, how they're potentially, are being used and hopefully will be used in the future to bring the next generation of cancer immunotherapies to market. And then kind of how GenScript can help, what we're doing to kind of support in this initiative. So with that said, I think the best place to start is to kind of go ahead and just give a very general overview. I'm assuming since the audience has come to a neoantigen talk that everyone is, of course, well aware of what the basis of immunotherapies are. For the record, there are two major groups of immunotherapies on the market today. There, of course, are checkpoint inhibitors, which are essentially ways of blocking different checkpoints and allowing you to kind of take the brakes off the immune system to hopefully help to get your immune system, or let's say a patient that's suffering from cancer, help them recognize the cancer cells and attack. Very effective treatment. It is not perfect. Like any treatment, is a second-line therapy behind, of course, traditional chemo. An alternative approach, although very similar, is, of course, CAR T therapy. Of course, TCRTs are in development. So CAR T therapy is really taking a patient's immune cells and then going ahead and training them, modifying them, such that they'll go ahead and they'll recognize a particular cancer epitope. And currently on market today, you have treatments like Kymriah and Yescarta. And of course, in the case of Kymriah, it's a recognition of CD19, which is overexpressed in cancer cells. So again, both approaches have their pros and their cons. What I thought was really, really compelling is a quote that I recently came across. So this is from a 2017 publication from Steven Rosenberg's lab. I'm sure everyone is well aware of the fact that Steven Rosenberg has been intimately involved in the development of immunotherapies. And so I'm not going to read the entire quote, but what really struck me is this. As it relates to melanomas, the large majority of patients with common epithelial cancers, which account for 90% of cancer deaths in the U.S., do not respond to immunotherapies now in use. So I just want to let that sink in. Billions of dollars, God knows how many hundreds, thousands of man hours. And we're making a dent in the fight against cancer, but obviously there's still plenty to do. There's plenty of new things to consider. There's need for additional therapies, along with, of course, the excellence in checkpoint inhibitors and with the latest CAR Ts and the soon-to-come to market TCRTs. So what's a way that we could approach this? What is an alternative way that we could improve the efficacy of cancer immunotherapies? And neoantigens are one of the potential targets that we could go after and we could develop therapeutics. And so what are neoantigens? So at their core, neoantigens are essentially variants, so SNPs, point mutations, indels. They're expressed or found in cancer cells, but not in healthy cells. And that's not entirely surprising here. We know from plenty of sequencing of biopsy samples how radically distorted and how, for lack of a better term, how messed up the genomic sequences of some of these cancer cells are. And so what you have is you have these mutations. They're expected to be very, very individual specific, tumor specific. So what that means is every patient is expected to have a slightly different neoantigen profile, for lack of a better term. And I want to point out that in terms of classifying neoantigens, what is one versus a tumor-associated antigen is something that we could talk about, and I'm happy to talk about in the question and answer section. But again, just for the sake of today's talk, think of neoantigens as something that you find only in cancer cells. It's something that's going to be different from patient to patient. If it's a true neoantigen, and what I mean by that, if it's an antigen that could be recognized by patient's immune system, it's obviously got to be expressed and then presented on the MHC complex on the surface of the cell. So a T cell can come around and say, hey, I see something here that doesn't seem right. What's really, really exciting are neoantigens, by their very definition, are not self-antigens. So they're likely to be highly immunogenic. And I'm sure everyone has a very good understanding of non-self and self as it relates to the immune system. Suffice it to say, obviously, self-antigens are not encountered by the immune system or are not readily recognized by the immune system for obvious reasons. So the idea that you have mutations that are only found in cancer cells, and potentially are highly immunogenic leads us to say this is a potential target for the next generation of therapeutics. And it's worth pointing out that this is a theory that's backed up by science. This paper was really, really interesting to me, so I cited this one. There's many of them. For the record, there has been at least a decade's worth of work where folks have gone ahead and looked at different patients that have cancer and have gone ahead and looked at the T cell profile of those patients. In other words, looked at all the T cells within the patient or within PBMCs extracted from the patient. And ultimately, it's well established that there are T cells that recognize specific neoantigens. So this paper comes from 2014, again from Steve Rosenberg's lab. It's a report of a single patient enrolled in a tumor infiltrating lymphocyte therapy trial, where ultimately this patient underwent sequencing of their tumor. At the time, the state-of-the-art was whole exome sequencing followed by RNA sequencing. Things have changed a little bit in that direction, but it's essentially the same technology as we are using today. And there were ultimately 26 non-synonymous mutations identified. It was found that a large percentage of CD4 positive T cells extracted from the patient were able to recognize several neoantigens, and specifically neoantigens associated with the ERBB2IP gene. So that's great. And so what the authors ended up doing, or what the clinicians in this clinical trial ended up doing, is going ahead and taking the cells, expanding them, and giving them back to the patient. And what they saw were quite a few things. I'm obviously grossly simplifying this paper. I encourage everyone to go ahead and to read it. It's very exciting. But two main takeaways. One, the patient showed robust interferon gamma response within their serum. Two, that ultimately post-treatment, 7 months post-treatment, they had a reduced tumor burden. So again, a positive sign. There's been more recent publications, but I thought that this one was especially detailed and very, very useful to kind of give everyone a primer. So again, we know that neoantigens are real. The immune system can recognize them. It's a matter of how do we encourage the immune system to recognize these neoantigens. So there are a lot of different neoantigen immunotherapeutic platforms that are in development right now. And this is, again, apologies of gross simplification. They really fall into two buckets. There's personalized cancer vaccines and personalized T-cell therapies. So for the purposes of today's talk, I'm going to focus more on the former and not so much the latter. If anyone is very interested about personalized T-cell therapies, I'm happy to talk a little bit about that at the very end. There actually is a role for peptides in that whole process, and actually for CRISPR as well. So I would definitely love to talk about that. But for the purpose of today's talk, we'll focus on personalized cancer vaccines. And the concept's pretty simple. The idea is that, okay, you've gone ahead, you have a patient, you've done whole exome sequencing or whole genome sequencing, you've established punitive neoantigens, and you want to then go ahead and somehow, you want to deliver those neoantigens into the patient. You want to either go ahead and take DNA, RNA, or peptide, and essentially make a cocktail of neoantigens that you think the patient's immune system is already starting to recognize. It just needs kind of a kick. And so you take, again, depending on the modality, Let's say, for example, it's peptides. You take a mixed pool of peptides and you essentially inject that pool of peptides, so the peptides are an active pharmaceutical ingredient, into the patient. And the idea being, if the patient gets this extra dose of the neoantigens that are already presented on their cancer cells, that they're more likely to recognize that particular set of cancer cells. You're going to have an expansion of T cells and a correspondingly aggressive immune response. So again, that is a theory, and it's based on pretty good science. So what I'm showing you on this slide is a really great paper from 2017 from Dana-Farber. This is from Patrick Ott and Catherine Wu's lab. Apologies, I know there's a lot going on here. I'll kind of set it up as best I can. So this was, again, preliminary clinical trial data. It's a phase 1 clinical trial. There were 6 patients that underwent this treatment as part of this clinical trial. They all had melanoma, and they all had neoantigens that were identified, again, by just genome sequencing. Ultimately, the group at Dana-Farber went ahead and not only did sequencing, but also some in silico prediction, some RNA-seq, and they did some screening, which we'll talk about in a second, and they came up with a mixture of 13 to 20 peptides encoding for neoantigens present within, slightly different between each patient. And what you can see on the far left is an example of an immune response assay, where, and just to give everyone some context here, what the folks were doing was essentially working with an ELISpot assay. Apologies, I believe it was interferon gamma they were measuring. I don't have that right in front of me at this point, but what you can see is going from the top to the bottom, patient 1, 2, 3, 4, 5, and 6. You can see the overall dosage scheme for the individual patients. And what I think you can ultimately see in different colors is a pool A, B, C, and D. These are different pools of peptides. So each pool has a slightly different combination of peptides, the idea being to go ahead to determine, okay, which peptides specifically invoke an immune response. You don't give the patient a single one and just wait to see what happens. You want to go ahead and give the patient as many options as possible to try to, again, invoke an immune response. What I hope you could see is ultimately this resulted in a pretty robust expression or release of cytokines. After additional tests, which again, I apologize, I can't get into given the length of today's webinar, what they ultimately found was a very, very robust CD4 positive response, not as robust as CD8 positive response, unfortunately. And then overall, 4 out of 6 patients showed no disease recurrence 25 months post-intervention. So again, another example on why I think the community believes that this is a potential avenue for that, again, next generation of immunotherapies. So very exciting. Since then, there's been a variety of different groups that have developed not just peptide-based, but also DNA-based, RNA-based, and I'm not as familiar with it, but also dendritic cell-based vaccines. So a lot of exciting things going on. So if we were to envision what an idealized neoantigen therapeutic development workflow looks like, it would look something like this. And apologies, I know this is, again, a gross simplification. What you're seeing here is, again, this idealized journey. And the way to think about it is it starts with the patient. It starts with taking a biopsy from the patient, doing sequencing, doing in silico prediction, and from that, you try to figure out, okay, what are the neoantigens that you think are most likely to invoke an immune response? It's worth pointing out that no algorithm is perfect, and currently I've heard estimates as low as 5% in terms of being able to predict a real neoantigen from a false one, from just a point mutation that happens to be in a particular cancer, in a cancer cell. At this point, you're going to want to narrow that list down a little further. And so that involves peptide production. And this is something universal, regardless of whether you're working on or considering working on a personalized cancer vaccine or a personalized T-cell therapy. There is this need for peptides. So you ultimately produce peptides corresponding to the sequences you identified in your sequencing in silico prediction. You then do some sort of preclinical screen using traditionally ELISpots, or I see a lot more groups focused on flow cytometry-based assays at this point. But the idea is really, okay, can you identify potential neoantigens, can you confirm that they're able to invoke an immune response and that the patient cells are able to, again, release cytokines to respond, essentially. If you're developing a platform, you might be doing some preclinical testing in animal models and obviously doing safety and toxicity studies. But assuming the platform is already been approved and that you're entering or about to enter a clinical trial, obviously at this point, you've done your screen, you know what peptides will work, at least in theory. And then again, you ultimately either, whether it's DNA, RNA, or peptides, create your cocktail, your vaccine, give to the patient. Afterwards, even if the patient, hopefully the patient does respond, there is a remission, you still need to go ahead and you need to do post-clinical screening. And the reason why is because ultimately, it's not just that you did an intervention, but that did the intervention work? Are you able to assay that? And again, that will require peptides. So you can see that neoantigen peptides, or I should say peptides that encode neoantigens, are critical for several different stages. Anywhere from initial screening. Also, by the way, they're also used in identifying TCR antigen binders, involved in efficacy screening after a treatment or after an intervention in a clinical trial. And depending on let's assume it's a vaccine, if you're developing a personalized cancer vaccine that's peptide-based, it can serve as the active pharmaceutical ingredient for that vaccine. So ultimately, this is great, but there's a challenge. And this is a challenge that's well reported in the literature. It is a challenge that comes regardless of, let's say, whether you're doing it in-house in terms of peptide production or you're outsourcing it. It's just a universally acknowledged fact that for a reagent that's so critical for this overall process, it's extremely difficult to get and to get in a timely manner. And again, I'm going to give a shout out to Catherine Wu's lab again. They came up with a very fascinating paper about 3 months ago that talks about their approaches to try to improve peptide synthesis, and they talk about the difficulty. Definitely a really exciting article to read, and I really recommend anything from her lab. We could go through a lot of details in terms of why peptides are difficult to synthesize that encode neoantigens. The three biggest characteristics that kind of make this difficult are length, hydrophobicity, and purity. Length, I think, is something that everyone understands. The longer it is for anyone to manufacture a peptide, the more difficult it is. And some neoantigen peptides are very easy to synthesize. For screens, you're typically talking, I have on the slide right now, 9 to 11 MERS. I've seen 15 MERS become 15 amino acids becoming more popular. But they can be as long as 40 amino acids, depending on the assay you're trying to run or if it's an active pharmaceutical ingredient. So the longer something is, the harder it is to make, the more reduced the coupling efficiency is, and so on. Hydrophobicity is a big concern. Not surprisingly, a lot of the mutations that go ahead and invoke immune response ultimately result in amino acids becoming hydrophobic. In other words, there's just more hydrophobic amino acids that are present on neoantigen peptides, and they tend to have a more even distribution of hydrophobic residues across those peptides. And again, this is all in comparison to, again, the approximately 10 years of work that GenScript's already done. So we, again, know that this segment of peptides is just very difficult. The last characteristic is purity. Then you might be saying, well, why does purity matter? And it matters if you are working with, let's say, a precision sample, like a patient sample. Obviously, you can't just go back to the patient necessarily and ask for another sample after they've gone through so many treatments. There's also, of course, the need for extremely pure peptides as it relates to, again, if we're working with an active pharmaceutical ingredient. For screening, it's not as important, but again, for some of the other applications or some of the other use cases, it is definitely difficult important to have a high purity. So all these factors come together to make it very difficult to synthesize these peptides. And this is something, again, that is well reported in the literature. It's something that our collaborators have talked to us a lot about. And so when we entered this space, we recognized this was going to be a pretty significant challenge. And so really, how do we solve this challenge? How do we go from we can do it, but not as well as we'd like to do, to something that's a little better? So where we're at right now is by using a combination of bioinformatics and chemistry approaches, we're able to achieve overall a 95% success rate in the synthesis of these difficult peptides. I want to point out we're only talking about neoantigen peptides. We're not talking about, for lack of a better term, run-of-the-mill peptides, which support projects that are not neoantigen-based. There we have an overall success rate of about 98%. So overall, we think that by using both these approaches, we've not only improved, but we'll continue to improve how we're able to deliver peptides. So I'm going to start with bioinformatics first. So one of the benefits of having taken on a variety of these projects just when this area of research was really starting to take off is the fact that we actually have a pretty good track record of not only what we synthesized, but candidly what we haven't always been successful with. And by collecting all this information, all this production data, we know what makes something difficult and what makes something easy. So as part of that, in data mining our production records, we've developed an algorithm we call NEOPRE. And this is an algorithm that allows us to just essentially by getting a sequence from a prospective collaborator or client to assess, is this essentially, for lack of a better term, simple, medium, hard? Is it easy to make? Something that may take a little extra, or is it something that we're going to have to put a little more work on, more resources on? All of these things we can take on in terms of projects. But overall, this algorithm allows us to come up with a score and essentially also guidance in terms of what should we do at this point, how we get around this. And so I'm showing you an example from one small study. You're looking at 7 peptides here of varying lengths. These are all neoantigen peptides. They come from different projects. There's no overlap from one particular lab or one group or anything else like that. You see a variety of parameters. I'm not going to get too much into the weeds, but what I draw your attention to is the last column. In the last column, the system is actually smart enough now that it'll kind of output some parameters and say, this is what we recommend you do. In other words, what the computer is telling our production team, based on what you did before, this is the way you should do this one. So I think it's a really cool thing. We're continuing to put a lot of resources in improving this. But overall, the idea is, again, we can take a list of potential neoantigens. We can then go through that list and we can right away say, okay, how can we best do this? And to give you a flavor for, well, it's an algorithm, great. How does it work and how accurate is it? Let's do accuracy. So for accuracy, what we're looking at is a small pilot study here, looking at 88 total peptides. These are peptides that just happen to fall within I want to say a 3-month window is what I want to say, a month or 3 months. It's just a couple of projects. Apologies, it's not as diverse a sample set as I'd like, but it's not a bad one. And ultimately what you can see is a combination of what's been predicted to be easy or difficult. This is an older version of the algorithm. And then what was ultimately shown to be easy and difficult. And what you can see is that overall the accuracy rate that we're getting is around 82%. So it's not bad. So more often than not, the algorithm does a pretty good job at setting the right expectation and then giving guidance in terms of, well, what is it going to take to accomplish this? How could we best do this? How could we manufacture this? Another more extensive data set. This one is from 3 months. You can see a total of 2,267 peptides. You can see our ultimate success rate, which is about, again, 95% for this 3-month window, and then overall 95%. And what you can see is a combination of difficult and easy peptides. You can see how many that we were able to go ahead and deliver. And obviously, not surprisingly, we have a small edge as it relates to generating easy peptides, which of course makes sense. They're easy to manufacture. And what you can see, of course, is what I discussed a little earlier, which is the longer a peptide is, on average, the more difficult it is to manufacture. But even there, we're actually doing pretty good. We're at about 78%, give or take a little bit. So overall, not bad. Definitely a place for improvement. But on average, when someone comes to us, approaches us on a project, we're able to go ahead and deliver 95% of what that client or what that prospect would like. Turnaround time is also an area of improvement for us as well. Obviously, since we're talking about personalized medicines, The answer to when do you need it is always, of course, tomorrow. We recognize that. And so we've got a lot of really great projects that are kind of churning internally that ultimately help to reduce that turnaround time. For now, though, you get a flavor for the turnaround time that we're talking at. And based on our market research, this is actually a pretty reasonable turnaround time for the quality material that we're providing. So the next part of this is really picking the right platform. It's going from, okay, bioinformatics, and figuring out what's best to make, how we're going to make it, and how it can occur several different ways. Obviously, we've got a lot of old school chemists that are part of our production team. We have an extension production team, first off, hundreds of folks. Apologize, I don't have the numbers right in front of me at this point. But last time that I looked, we had a huge number of not just technicians, but also scientists and quality folks to ensure that we continue to produce quality products. In terms of the actual science here, we're really using three major platforms. We're using microwave technology, liquid phase, and solid phase chemistry. In general, microwave technology tends to be the platform that we default to. For most of the projects, it's automated. It gives a pretty good yield. The yield involved in neoantigen peptides is actually kind of small. It's in the milligram quantities. For screening, it's honestly even less sometimes, microgram quality. Turnaround time is pretty good. We also have traditional liquid and solid phase chemistry. And obviously, we have a wide range of synthesizers, HPLC equipment. The way that we really structure our production service is really to be that Swiss Army knife, to be able to take on just about anything that comes our way. And one of the ways that we're continuing to kind of stay current with the market and obviously continue to build on our leadership position is through investment in automation. So what I really want to show you in this slide is what you can see on the far right here. This is our PEP HTS library platform. This is an extremely large automated synthesizer. I kind of feel bad having the picture like this. It's a little small. I've had a team from our production site go ahead and kind of give me a walking around tour since, unfortunately, COVID-19 is preventing me to see the new platform. But it's large enough that you can spend a good half a minute going in, just doing a whole circle around the thing and get to the point that you started at. Able to synthesize 600 sequences at a time and do it in 24 hours. Again, we're working on new forms of chemistry to go ahead and to improve this and to again, accelerate the turnaround time. Ultimately, we also have capability for producing large-scale peptides. I'm not going to talk too much about that because, again, that's kind of outside of the scope of today's talk. And for most folks, milligram quantities are sufficient for neoantigen peptides. So one of the newer things that we've also got going on, it's again, not just the equipment, which is obviously fantastic, it's also the chemistry itself. So with our high-syn chemistry, and this is a new coupling chemistry, but it's also not just, I should say, just the chemistry, but it's also the production process, the platforms that we're using. A lot of great thing goes into making a new chemistry. What I'm showing you here is proof of concept data. So if you look, there are two peptides that we would consider difficult and have found to be difficult. What you can see on the top row is our approach, or I should say our effort to synthesize those peptides using our standard process. And what you're looking at, of course, are Uv-Vis traces. And what you can see are the different peaks indicating something, not necessarily something that you're looking for, the peptides you're looking for. In the case of the blue arrows, those point to the peaks that we are looking for. And you can see that using the standard approach, again, apologies, small sample size, we're only able to produce one of the two peptides. And again, I want to emphasize these were difficult peptides to make, and obviously we expected this. However, using our new high-syn chemistry, what you can see is not only were we able to synthesize the peptide, what really struck me as interesting about this is how little excess contaminant or side product is present within the trace, essentially. And obviously, that's just going to make it that much easier for us to go ahead and to get a higher purity for use in clinical assays. So again, ultimately, what we found is that this approach works and should add value. So what does that really look like? So in terms of numbers here, sample size of 232. So ultimately, using our new high-sen chemistry, we took 232 peptides that we weren't able to manufacture before. And of those, we were able to synthesize 200 peptides and get an overall synthesis success rate of 86% for that batch that previously we failed to synthesize. So again, it's not our success rate overall is 86.21%. It's a matter of for something we couldn't manufacture before, we're at now 86%. So that adds on top of the 95% I talked about earlier. You can see another example of that. This was, I want to say, a data set from a couple of months ago. Actually, I take that back, about 4 months ago. And in this case, we're working with 88 peptides and ultimately able to synthesize 80 of them. So we continue to improve the chemistry and tweak it. And again, it's not just the reagents, but it's also the overall process. So by making this kind of investment in both platform and also within new chemistry and new processes, along with investment we obviously have in our employees and our people that do the work, we're increasingly getting better and better able to make these difficult peptides, and we're one of the leading providers in the market today. So it's not just synthesis, so it else goes into that. Anyone can, there's a lot who can make a peptide, but it requires more than that. So if you're going to ultimately use these peptides in even a screen, one could argue, you're going to want to go ahead and you're going to want to do different quality controls. And especially when you think about ultimately assuming that your ambition is to generate a peptide-based personalized cancer vaccine or some sort of peptide-based therapeutic, you want to control for a wide range of different candidly processed contaminants. And also because it's a requirement of the US FDA that you know exactly what is going into your active pharmaceutical ingredient. There are a variety of tests and QC options that we have to offer. I would point your attention to, endotoxin control. Obviously, wanting to have low amounts of endotoxin in any peptide is useful. Even if you think about it from the perspective of a ELISpot or any sort of T-cell activation assay, endotoxin itself can invoke a non-specific immune response, and so it's something that you obviously do not want to have in your material. But there's also tests that you want to do in terms of validating the identity. So, of course, LC mass spec. You're going to want to go ahead and peptides are great, but let's be honest, they're not all made equal. They're not all equally soluble, unfortunately. So doing solubility testing, that way you're protecting the investment that you've made and you're able to get value out of that. And again, I could go line by line, but the overall, what we offer at GenScript is a really complete package in terms of the quality material, and we customize that. Increasingly, as we move beyond RUO grade material to preclinical and eventually to GMP compliant material, we'll obviously go ahead and tighten up these specifications and start to rigorously suggest and very, very strongly suggest for specific requirements based on our experience. And that'll be kind of a value add for you when you're in the phase of wanting to consider peptides for these applications that are, let's say, more clinical. Another thing that we offer is we offer library design tools. So I mentioned earlier some of our bioinformatic tools. In addition to that, we've obviously got very simple tools that allow us to go ahead and to build, let's say, overlapping libraries, for example, as you try to kind of narrow down specifically where your epitope of interest is. We're also collaborating with a variety of different companies and individuals to try to improve our ability to add value in terms of identifying neoantigens. Recently, we've entered a collaboration with Cayman Chemical, which is involved in immunopeptidome screening by way of mass spec. And so they're ultimately identifying punitive antigens, punitive targets, and as part of that service offering, they're recommending our peptides for use in validating those potential hits. So there's a lot to do. There's a lot of exciting things to talk about. At this point, I just want to very quickly give an overall overview about what we have to offer. Again, what we have to offer for neoantigen therapeutics are really peptides that are used for screening, are used for post-clinical efficacy testing, And then ultimately, again, our ambition is to be able to start to provide active pharmaceutical ingredient quality or GMP quality material for the use in personalized cancer vaccines. Ultimately, I think what you can see from what I've talked about, and I'm happy to talk about some of our experiences, is that ultimately we've been doing this for several years at this point. We've got a very well-established track record. We've been cited in quite a few publications. We have quite a few collaborators and folks that we've worked with over the years. Overall, we're able to achieve a high success rate for the synthesis of these difficult peptides. And at the same time, we're able to give you that flexibility. I mean, GenScript is focused on flexibility, customization. That's something that will continue to be part of our DNA, unintended. But again, we are also not only giving you that Swiss Army knife, but also starting to give you higher quality material with more tightened specs that will ultimately support you not just in your research, but also in your development and then your clinical assays. So with that, I want to go ahead and thank everyone for their time. Appreciate you taking some time out of your morning to hear me speak. And with that, we will go ahead and we will go into question and answer. So thank you very much.